Hi guys, today we are covering snake anatomy. So what you can expect to get from this video is a general understanding of snake external and internal anatomy, as well as a understanding of the differentiation between the two families of vipers and colubrids. So first, in order to better understand these families, we're going to go into the phylogeny between the two. First, we're going to start from tetrapods. From tetrapods, we get two branches. The branch that we're concerned with is amniota, which are known for the animals having an amniotic sac. From there, we're going to follow the cladogram down to Lepidosauria, which the animals in that are known for all having overlapping scales. From there, we're going to jump to a new clade called Toxicifera. Now, this is a relatively new discovery, and it's still a little bit hotly debated among scientists currently, but it shows the presence of a potential venom system and a certain protein that's produced in the oral glands of its inhabitants. While not all members are venomous, they do all produce this protein, and this includes some lizards as well. From this, we are going to go down to the next division, which is Cainophidia. And now this contains all of the snakes that are considered venomous. It's also worth noting that not all of the snakes in this classification are venomous, but all the ones that are venomous do fall under this. So now the first branch off of Cainophidia that we are looking at, and the most primitive branch, is the vipers or Viperidae. So they are known for having long, hollow fangs that are specialized for deep injection of venom. And this is the family that our rattlesnake that we'll be dissecting belongs to. The other clade, which branches down and then goes off again, is the Colubrids or Colubridae, which is known for its members having rear-facing fangs. And this is where our non-venomous snake belongs to. It's also worth noting that not all colubrids are non-venomous. Approximately 50% of colubrids are considered venomous, and the rest just have regressed venom systems. So now that we've discussed the phylogeny a little bit, and you understand where these snakes are in relation to each other, I'm going to hand you off to Lauren to discuss the dissection and the internal and external anatomy of these snakes.
So these are our two snakes. On our bottom, we have our venomous rattlesnake. This is family Viperidae. And on our top, we have our colubrid. This is the non-venomous snake. So there are several external anatomy differences between colubrids and vipers. One of the first ones is the heat pits, which are located on the face of the viper. These aid in detection of prey during hunting. On our colubrid, we do not have these heat pits. They find their prey via other senses. Another difference between the two snakes is the vipers have a vertical pupil, while the colubrids have a round pupil. These snakes can also be identified by differences in dentition. As you can see, this viper, which has had its fangs removed, they typically have long, hollow fangs to inject venom into their prey. However, the colubrid has backwards-facing hooks for teeth, which are not usually hollow for the injection of venom. Another external feature which can be used to identify these snakes is the general body shape. As you can see, the venomous snake is a lot skinnier and flimsier, while the constrictor is a lot thicker and more muscular. The final external difference between these two snakes is the postanal scales. As you can see on our colubrid, we have the anal plate, which is where the cloaca is, and the belly scales. Before the anal plate, it is a single scale. However, afterwards, it becomes two scales. As you can see, cranially, it's a single scale, caudally, it's two. On our venomous snake, we have the anal plate, while we have a single scale, both cranial and caudal to the anal plate. Another external feature that all snakes have is a kinetic skull. As you can see here, the dentary bone and the cranium are both non-fused, so they can be moved around which will accommodate for consuming large prey. To dissect these snakes, we did a ventral slit along the side of the body, just so that we could fold it open without destroying any large features. So this is our non-venomous snake, and we are using this for our representative snake. As you can see here, this below the tongue, this small opening is the glottis. This connects to the trachea and is used in respiration. This is the esophagus, which is highly elastic to allow for swallowing large prey items and moving them to the stomach. This small tube that we are pulling out is the trachea. It is formed by small cartilaginous rings which are not entirely fused, so it's more of a C shape. So this is our heart. This is the ventral aorta down here, and this is our heart. And then this is our vestigial left lung. And we also have our larger right lung. As you can see here, the vestigial left lung would normally be larger in other tetrapods, however it is reduced in snakes. Then we have our esophagus leading into the stomach, and this is the thyroid gland right here. Then we move to our liver. So this is a very large liver, takes up a lot of the space within the abdominal cavity. And then caudal to the liver, we have a large deposit of fat. This is very similar to the greater omentum in mammals. Hidden below this fat, we have the gallbladder and the pancreas. The gallbladder is located directly attached to the liver and is this green bit here. And then right below we have the pancreas, which is fused to the caudal end of the stomach. Underneath all this fat, we go into the lower digestive system. So right here we have the small intestine, which is bound by a mesentery, and our large intestine. The large intestine is relatively small in snakes because of their diet. Next to our large intestine, we have our two ovaries. So these are the paired reproductive organs on this female snake, and they connect to the cloaca via oviducts. Here we have the large kidneys, which take up a good portion of space in the caudal end of the snake. This is something really interesting that we found in our non-venomous snake. These are parasitic worms, which as you can see were harbored within the lung of the snake. You can actually see two small holes where they had anchored. A final internal way to distinguish between colubrids and Viperidae is the location of the vestigial left lung. On the top, you can see that the vestigial left lung is located caudal to the heart, while in our viper, we can see that the vestigial left lung is located anterior to the heart.